Hello everyone and welcome to AP Biology with Mr. Jennings. Today we're going to talk about the most single complex group of macromolecules, proteins. Now proteins, like I said, are the most complex, and they were so complex at one point in time we thought they did everything including our heredity. Turns out we were a little off from that in science, but the complexity is the reason why. First off, proteins can be made up of carbon, hydrogen, oxygen, nitrogen, and sulfur. First group to actually see the sulfur and nitrogen here. And it does about everything in the body, again, except genetics. Now, it's heavily involved in genetics, as we'll learn, but it itself is not the genetic material. Now, proteins, returning back to the normal nomenclature here, do have a monomer. That monomer is the amino acid. We'll talk more about structure here in a minute. And monomers, these amino acids can be linked together via peptide bonds. Often this is where the name polypeptide starts coming in. So if you are familiar with a little bit of chemistry or anything like that, you may have heard the word peptide. That's where the name Pepsi gets its name. Pepsi used to have an enzyme in it known as pepsin, and it was sold as a stomach relief, a stomach medicine type thing to help you digest. So pepsin is a very valuable enzyme. It breaks down peptide bonds. And multiple peptide bonds make, well, proteins. So let's talk about the amino acid structure here. All amino acids typically follow this basic structure. There's currently about 20 or so majorly identified amino acids. That number does fluctuate between different sources, but safe to say about 20 of them. Now, amino acids all follow the same basic group here. You have an amino group. Now this amino group is an amine functional group with a hydrogen extra in there. That allows it to be a little bit more positive, and it gives it a little bit of the unique chemical properties, where it gets the acid terminology here. We have a carboxyl functional group. Carboxyl functional groups are noted for having a double bonded oxygen and a single bonded oxygen to a carbon. Backtrack here. Amino groups are known to be kind of like ammonia. It's going to be nitrogen with 3 hydrogens on it, or sometimes just 2. You don't see this plus every time. And there's going to be a basic hydrogen just attached there to the carbon. Off that central carbon, you're going to find what we call the R group. This R group can be any functional group. And this is where the complexity can happen. So there's 20 different amino acids to make 20 different functional groups bond to this R site. When we have this active site, or sorry, not active site, this R grouping, anything can take its place, and that's where we're going to get a lot of variation. And not only does it give variation, the chemistry of that R group can really affect the overall structure of your protein. See, proteins have 4 levels of structure. There's your primary structure, and this is pretty much your polypeptide bond. This is where all the basic bonding takes place. You got yourself an amine group here, central carbon, carboxyl, and it's going to bond these little different R groups. Nothing special. Pretty basic. Now, the secondary structure is typically what we call, where we get our alpha helix and your beta pleated sheets. That's typically what's associated with this group, this level of structure, and that is these interactions between the functional groups. So we have what we call hydrogen bonding and other intramolecular forces. Now those can be stuff like a disulfide bridges, types of hydrogen bonding, different other types of intermolecular forces. Because they interact with one another, the protein's going to start twisting and folding in on itself. So the R group matters. Now, each R group has a unique chemical property, and each R group can be lumped into a category of similar chemical properties. So if the R group's similar enough, you may get enough of this bending and folding. Now, once you have enough bending and folding, you're going to get what we call a tertiary structure. And that's the overall big picture. It's when the overall interactions between the R groups of this side of the protein and those R groups, and it just gets a big overall big picture of a twist. Then there's quaternary. Quaternary is large. Quaternary is typically multiple proteins interacting with one another. So a good example of this is usually hemoglobin, or any kind of heme groups, as we see here. This is what this is. This is a hemoglobin. That's what the protein structure we're seeing here is. See this heme. Hemoglobin is one of the most common blood molecules. It's a molecule inside of red blood cells that helps carry oxygen and CO2, amongst other things, to your bloodstream. It's a very complex molecule. Now, quaternary structures are not that unusual, but we don't tend to talk about them too much because it gets a little complex, and it's beyond the scope of the course. Now, when it comes to protein structure, now I've talked about this when it came to icebergs. Protein structure affects organismal physiology. Now, what does that mean is form dictates function. It determines it. If the form is not correct, the function is not going to be correct. This is a very important statement. I'll say this a billion times in class. Form determines function. So the shape of the protein is determined by the chemistry of the protein. Change the chemistry, meaning change in our group, you change its shape. Changing its shape means you change its function. A change in genetic code can change the genetic chemical properties of a protein. So if I change just one base of a nucleotide, I could potentially change an entire protein. A great example of this is cystic fibrosis. One single protein change. One single protein change results in a completely different functioning protein in your cell membrane. Sickle cell anemia. Sickle cell anemia. One change in a protein changes that hemoglobin into a different shape. Because it changes hemoglobin into a different shape, it changes the shape of the cell, and the cell has a totally different function and it cannot carry oxygen as efficiently. Now, other things you can do, not only can you change the genetic code, you could denature it. Stuff like high temp, pH, salinity. All these things here can actually cause a protein to change its shape because it messes with the chemistry. When you have different chemical interactions going on, it's going to cause the protein to morph. So high temperatures, some proteins work efficiently. High temperatures, some proteins do not. They kind of fall apart. Same with pH. So a lot of proteins have what we call an optimal range, an area where things work the best. And if you change that, if you follow that optimal range, they work, but not as well. All right, let's wrap it up here. So proteins are probably the most complex and the most important macromolecule we talk about in this class. We spend a good number of our time on it. We talk about a numerous amount of enzymes. We talk about enzymatic functions. We talk about other reactions. We talk about all sorts of things. A majority of the stuff we talk about in this class is protein. Now, that does not mean protein is the only one that's important. It just happens to be the most complex. Because it's so complex, we used to think it was genetic code, and it turns out it wasn't. It's one of the few things that's not in genetic code, oddly enough. It's involved in the process, obviously, and it's a result of genetic code, but itself is not hereditary. So it does a lot of things like structural support, it does cell transport, it does immune defenses, it does a little of everything. So proteins are so important, and one thing we're going to spend a good number of time on. Alright, so that wraps up this video. So the next one will be the conclusion of this little series here, and we're going to talk about nucleic acids, probably the simplest of all macromolecules. Alright, thanks for watching. Keep in touch. Adios.